Okay, so today I would like to present part 2 about predicting the brain. What's next? So I have decided to continue with the plan of today to discuss network dynamics, what is our effort and the effort of many colleagues of reconstructing neuronal networks from time series. So this remains an open question, not very well solved, a lot of discussion and issue about the stability of the reconstruction, and behind this the question is how the brain use neuronal networks. Are there paths that are encoding information or just neurons, single neuron that has been the dogma since, I would say, Hubel and Wiesel discovery. But maybe in paths there are information hidden that is not clear. And then I would like to move to brain oscillation, basically based on the EEG and what is the trends in predicting the brain and how we call it today predictive medicine, what it is all about. And not today, the next time I'll discuss this nanocommunication. We will go more into physiology, discussing nanocommunication between the neuronal and glial networks. So, just to make a long story very short, there are three types of cells, mostly important cell in the brain. There are the blood vessels that are bringing the bloods, providing glucose and oxygens, and this is taken mostly by astrocytes. All of these components and are redistributed through the extracellular space and to neurons. And we have seen most of the communication between neurons at synapses, with the glial component having playing a role of regulation, but there are much more of these domains, such as astrocytes and feet, that communicate between cells. So the idea that has been prevailed over the last 10 years is that now neuronal networks that are excitable, that are important for neurotransmission, are really sustained by astrocytes, providing metabolic substrate, which was the classical view, but also participate to the excitability, synaptic regulation, that is the strength of the synaptic current is now known to be regulated by astrocytes. And astrocytes also regulate the excitability by controlling the potassium, the extracellular potassium. And we have a recent paper on this in Cell Reports for those of you who are interested. So the difficulty of studying the two networks is that if you patch a cell, if you patch neurons, you see that neurons are excitable. That is, they respond to an electrical current, while astrocytes stay mostly at minus 80 millivolt when patching the soma. So we'll see in the future that is not true everywhere. So that's why the role of astrocytic network has remained difficult to explore. And most of what is known comes from a neuron, although there are now understanding of this astrocytic network activity. So I would like to spend 5 minutes of how it is possible to construct hidden connectivity and what are these missing of this notion of hidden connectivity. So a lot has benefit from what is called two-photon fluorescent calcium imaging, where you have an indicator called a GCAM that reports the activity of neuronal astrocytes. So this is an example of a movie where you can see here the activity reported on astrocytes. Sometimes you see this activation, this white bleaching, this white transient responses, which is a reporter of calcium activity. And this occurs at different places. This is astrocytes in the slice, where you see this random activity. There is no specific activation here. So the question that we've been working, and many people have been working also on that direction, is if you see such a movie, what can you say about how astrocytes are really connected? And so how could you address this question? I mean, is it possible that through, let me repeat this movie again, is it possible that through this correlation between, let's say, these astrocytes here that where you see these calcium responses, and let's say that one, can you say something about the specific connection between, let's say, this one, and that one, and possibly, you know, the ones that are below? So in principle, it should be much easier than in neuronal cells, because astrocytes are mostly connected by passive gap junctions, so that the activity of calcium could be simply diffusing through these neighboring astrocytes. But the question is then, if they are really connected to hundreds of astrocytes, at least in the neighborhood, why only a few of them are activated? So this remains mostly open. This is in contrast with neuronal activity, where we know that there are long-range connections. This has been explored anatomically. So we know that if one neuron is activated, it will activate a neuron where you have the dendrite projecting or the axon projecting, so that the dendrite of the neuron that is connected will be possibly activated, leading to a communication between these two neurons. So in the case of astrocytes, we recently looked at it. That is, all astrocytes in principle that are connected to one given astrocyte should be activated under calcium, let's say, response from this astrocyte. Is it true that all of them are activated? Obviously not. And can we say something about at least some preferential path, although the exact mechanism can necessarily be known or clarified at this point. So the idea is that maybe one astrocyte could be activated, followed by another one, a third one, and so on. So you get possibly a path here. And if we have a path, the question is if this path can be used several times. In other words, is there something specific in this path, or is it just a random path? It could be one time it goes this way, another time it goes another way, and so on. In that case, there is no much meaning, no much information encoded in the past if they are not repeated under specific conditions. So now let's ask the same question for neuronal activity. So we recently looked at it, and you can isolate here region of interest which can be identified as, let's say, single neurons, and you can look at the response in terms of calcium imaging for the different voxel. Here we are in 3 dimension. And then one trend has been that if one neuron activates always before another one, and if you can repeat this across different section of activation, that is, you record several movies, then you create a link between this neuron, the pink one and the blue one. And then you can do this iteratively, so that in the end you can go from neuron 1 to neuron 2 to neuron 2 to neuron 3, as you can see here. And if there is a signal, it's not just noise, then across recordings you should find this path used again and again and again. This leads to an activation. And so we are currently investigating how much information there is really in this, or is this simply an artifact of just communication. It communicates through these paths, but these paths have no real encoding relation. They have no identity in the encoding. They are just propagating the information. And so just by cutting the signal at the minimum intensity, we're trying to look during all these segmented events. So this is segmenting an event at the global event that you can see here. And so each time you go to the minimum activity, you say, let's start again. And during this activation, we ask what are the neurons that have been correlated in time, so that you can generate a graph in each of these sub-regions, again, segmented in time. So you can generate this different graph and see how many paths are really an intersection of these different events, so that in the end, you reconstruct a graph that accounts for the amount of time two neurons were firing with a short delay interval. So those are called functional connectivity. This has been, studying for the past 10 years now. We are moving in vivo, where we try, and also in 3D, where we try to see if there are subnetworks that are more relevant than others. And by doing so, you can threshold according to the number of link, thus the number of time two neurons were correlated. And what you see, if you look at this specific region, we see that there are neurons that are indeed highly correlated in terms of the number of time one neuron fired after the other. And so you get here subregions that are not necessarily appearing when you just label neurons in terms of anatomical connectivity. But as functional connectivity, it reveals subnetworks that needs to be more understood to see why they are more connected, and are they containing some kind of encoding specific regulatory mechanism. So this is still unclear, and this will be very important to better understand how the brain encodes information and create subnetworks for this purpose. So this is now something that I have shown last time, is that on the bottom part here, you have what is called the raster plot. Each point corresponds to a firing of a single neuron recorded by an electrode here, where you see the tip. And you can, I said last time, you can distinguish neuron coming from the thalamus with respect to neurons that are in the cortex. So you can see the different type of spiking and where they're coming from. But what it is interesting is that you can at the same time record the local field potential, which mirror image the electroencephalogram, the EEG. So you have here these events here that are bursting events that now we can identify actually what are their neuronal origin. So now I would like to focus more on this coding of what you see in the LFP, that is these speedball events. And for this, I would like to talk about brain during anesthesia. So as you know, I'm sorry for this, it's perpendicular, but you still understand that you are in the surgery room, you have the surgeon, and while the surgeon is performing any type of surgery, the patient is usually sedated under anesthesia, and this is monitored by electrodes that are positioned on the scalp, usually on a different types of machine that reveal with this number here the level of sedation from 0 to 100. And to have somebody optimally sedated, the number should be between 40 and 60. This is a recommendation, and of course there is a lot of discussion about the reality of this optimization procedure. So we're actually currently interested in measuring the depth of anesthesia and to predict the arrival of deep anesthesia that could cause on the long term delirium, and also triggering all kinds of brain consequences, such as neurodegenerative diseases. So if you're interested, you can look at SignalMed Plus, where we develop this type of algorithms. So over the years, there have been understanding of what is in this electroencephalogram signal, where in the awake behavior, you can have this beta very fast activity or gamma activity that is indicative of awareness. However, in conscious, start with slow and alpha oscillation here, where you have the slower activity. First of all, you have these delta oscillations here, which are very slow, of the order of 0 to, let's say, 4 Hertz. And you have also this 8 to 12 Hertz, where you have 12 peaks here per second. And so this is an example of these slow oscillations. Of course, it seems that there's a motif associated with deep anesthesia which occur when systematically the dose is increased, which is called burst suppression, where you have a motif where you have no much of an activity followed here by this burst. And at extreme, when you continue increasing the hypnotic concentration, you reach the level where the EEG is locally flat for some long time period, which is called isoelectric suppression, which, if it lasts forever, is a signature of brain death. So you have this motif and you would like to understand how to read them. What exactly is the information hidden here? So this is still an open question. We still don't know about it. But so I would like to discuss now how this motif can be generated and how we see them today. So again, I'll show you example of recordings. Sorry for this rotation. So you have here the EEG, and during anesthesia, you can see that you have this type of responses, but sometimes you have also this burst suppression that occurs, and they are not very, they are often, they are not rare at all. And the question is how to avoid them. So now I would like to discuss the analysis of the signal itself very briefly. You usually use on the EEG a time window that lasts capital T in duration, and so you use this time window to develop what is called the classical Fourier transform, where you look at where is the power of the oscillations. And to compute this in this sliding window, you just use the Fourier transform. And for example, here you have the classical trends of the decay. That is, the amount of power for slow oscillations is usually the highest. That is, the energy of slow oscillation is the highest. And then it decays with a certain slope that could be characterized. And often here you have a peak at 10 Hertz, which is the manifestation of the alpha band. That is, you have, if you count here in 1 second, the events where you have between 8 to 12 peaks, this corresponds to these ensembles. And so you have basically 2 peaks. One peak at this delta band, this corresponds to the slow oscillations, and the alpha band here, which corresponds to this 12 Hz oscillation. And then you have a decay, and here we enter in the noise after probably 30 Hz. So when you collect this power spectrum, it really tells you where is the energy of the signal. So you get here this type of representation, which is called the spectrogram. And you go from as a function of time. You see very well this band here, the red band, which corresponds to the peak, while you have here the alpha bands, the delta bands, which corresponds to the second peak here. And then everything degrades as a function of the frequency. That is, you have less and less power. So the question is, how much information is there predictive power in this spectrogram? So over the years, it has been shown that it's very hard to make predictions. It's a good representation of the history, but it's a very bad representation of predictions. For example, if you're here, can you predict what will happen here? So in general, this is not easy. It depends on for example, the level of sedation, the patients, and all kind of activity. For example, if the surgeon, for some reason, has to cut specific tissue, it can happen that it generates a response in the brain. And so this cannot be anticipated by just the brain itself. So it has been very hard to use this as a predictive anticipation tool. So then the question has been moved toward this spindle activity, where you see these bursting events, where there is a phase of increasing for the envelope, a phase of decreasing, which is basically due to the communication between the thalamus and the cortex, where you have in the thalamus a projection to the cortex, which goes back to the thalamus, generating a positive and then a negative feedback loop. But what is interesting is that the general notion of having a positive activation followed by a negative activation, like say excitatory neurons that are exciting inhibitory neuron that feedbacks into inhibition, this actually contains the general mechanism for spindle activity. And I would like just briefly to mention this. A paper is coming out soon on this, that you can model this using what is called an Holcman process, where generically the matrix A has a focus with a decay of amplitude R and oscillation omega that is perturbed by a Brownian motion. So with this 3-parameter model, it's possible to obtain this spindle type oscillation characterized here by this alpha band, where you see here the peak at 10 Hertz, which is called the peak of stochastic resonance, that characterize this burst of activity. So what we are saying here is that basically the activity coming of spindle is simply the result of stochastic events between the thalamus and the cortex. And this the paper should come out soon as a preprint. So spindle, as we said in the conclusion, are generated as a stochastic resonance effect, and thus the conclusion is that there may not be any type of information in the spindle itself. And this answers long-lasting questions. Let me read this paragraph to you. I think it's important. This was the view of Lopez da Silva a couple of years ago on the pacemaker concept. In our view, this problem cannot be solved by using deterministic pacemaker concept. That is, if you want to understand what generates those spindles, it's not a deterministic neuron that will fire rhythmically. In a general sense, a deterministic pacemaker has two main properties. It is autorhythmicity, and it can impose its rhythm upon other systems. This implies absolute correlation, that is correlation of one or nearly one, if it is assumed that there is measurements of noise in all recordings. According to Lopez da Silva, alpha rhythm should be considered as signal produced by stochastic processes. More specifically, thalamic and cortical alpha rhythm would result from the filter property of neuronal networks when submitted to a random input. So this is the direction that we have been followed in the last couple of years, and I think these Holstein-Uhlenbeck processes shows that actually you can define and you can generate this alpha rhythm through these stochastic processes under a deterministic rules that you have excitatory neuron that activate inhibitory neuron that inhibit then this excitatory ensemble of neurons. And this is enough to generate this stochastic alpha process, alpha band. So in that case, there is little information in single spindle. And so this is an example of general anesthesia. You can see here the spindle. This is this spectrogram, and you can see here this 10 Hertz. This is in the log scale. This is the bump at 10 Hertz, and we have identified here this alpha spindle. That is, if you filter the signal between 8 to 12 Hertz, you have these beautiful spindles here. And now that we've understood that where they're coming from, we don't think there are much information in them. They are more representing what is really happening at the network level. So it's normal that they don't have necessarily the same length or the same amplitude because they are generated as a random process. So the conclusion is that the alpha band is composed of a succession of spindle making sentences. That is, probably if it's in the spindle itself, there's not much information like in the letter. You see in the letter, in each letter of a word, there is not much information, but the combinations is where the information is. So that was our view of spindle that can make sentences. And actually nowadays, if I ask you between the left and the right, which one is the true one and which one is a simulated one, it's now becoming very hard to distinguish. Actually, even myself, I don't remember. Showing that we have now good tools to simulate the spindle activity. So if you look at the representation of the brain during classical brain problems such as migraine, epilepsy, or transient ischemia, you still see this band of activity. And it remains quite difficult, actually, from the spectrum itself to classify in one of these three states. It's actually very hard, and it means that more information should be used rather than just the power, the transient features, contained in the EEG. So I will end here for this direction of trying to look at the brain through coding, what is coded, where things are coded, and what are the tools to decode. Machine learning itself or deep learning is very hard because sometimes we have no idea where the signal is. How to extract the signal, what's the difference? So the direction that we have been proposing is that we need to go back to the physiology and understand at the different scaling level where is the signal. So I will end with this picture where we try nowadays to bring art and algorithm. You can have a look at our website where we started to post some images about the organization of cells during brain development. All right, thanks for your attention. So I'll see you on the next video, part 3, about more of the nanometer level exploration of the brain. Thanks.